Welcome back, students. Welcome to Priya's Life Science Academy. This is your educator Priya. I hope you have watched the previous video and have understood the concepts very clearly. Today we will touch one very important part, that is the types of alleles. Okay, the first one is the pseudoallele. By the name you can understand that what can be pseudoallele. They behave as they are the alleles, but actually they are different genes. They are not the alleles of a same gene. When two types of alleles have similar function but different structure, okay, that is, for example, this is a chromosome, this is one locus, this is a allele, here is another locus. Okay, so these are known as pseudoalleles. Why? Because their functions are related. They perform similar type of function. They are of two different gene. You can see here, different gene. So different locus, but they function alike. Their functions are similar. They are functionally related. Okay, but they are alleles for two separate genes that contribute to the same trait. They are functionally related and they contribute to the same trait, but they are alleles of two separate gene. They are alleles of two separate gene. Okay, that is the difference. You see the diagram. By the diagram you can understand it very clearly that their positions are different. They are the alleles of two different gene, but their function is similar. That is why they are known as pseudoalleles. Okay, so when two genes with similar functions located so close to each other on a chromosome, they are known as pseudoalleles, and their positions are also very close to each other. That is why they are genetically linked. Okay, it can be the result of gene duplication events. It can be the cause that there is a gene duplication, that this gene is duplicated and has given rise this gene. So it can be the cause of pseudoallele, the presence of pseudoalleles. But the main thing is you have to understand that the pseudoalleles are the alleles of two different genes, but their functions are similar and they are very closely located. That is why sometimes they are inherited together as well. Okay, there is no recombination between them. As they are very closely located, there is very less chance of recombination. So they are, in most of the cases, they are inherited together. I'll come to this recombination inheritance linkage in later classes. Okay, so what's the example? In case of lozenge locus, what was once thought to be a single gene with multiple alleles was found to be a cluster of closely related genes. Okay, so it can be the notion that we can think that there's a locus known as lozenge locus. We first thought that it was the result of multiple alleles of a gene, but no, we were wrong. Then it came out to that they are different genes, but they are found in clusters. They look like they are the alleles of similar genes, alleles of a same gene. That is why they are known as pseudoalleles. Okay, so lozenge locus is a very important example of pseudoallele. In case of Drosophila, you can see this is the wild type eye and this is a lozenge eye. Lozenge eye means diamond like shape eye. The eyes shaped are irregular in this case. So lozenge is a locus, is very important locus which control the various aspects of eye development, where there are multiple alleles of different genes. So what is the functionality of this locus, lozenge locus? It's a composed of multiple functionally related but distinct genes. Okay, the first point of pseudoallele, that they are functionally very related but they are distinct genes that control various aspects of eye development. These genes are so close to one another that they are almost always inherited together, mimicking the behavior of alleles. I told you that their position is so close to each other, you might think that they are the alleles of same gene, same, but they are different genes. They are the allele of different genes. Their functionality is similar, but they are distinct genes. Okay, so this is an example of lozenge locus. In Drosophila, there is a locus known as lozenge. This locus is important and responsible for Drosophila's eye development. Whenever there is a mutation in lozenge locus, the wild type eye becomes lozenge eye, that is the diamond shaped eye. But in lozenge locus, there are a lot of type of gene with lot of alleles. They are not the alleles of same gene, they are the alleles of different genes. Okay, so despite being separate genes, recombination between them is rare due to their proximity, as they are very closely located. So recombination is very rare event. They exhibit genetic linkage. They are always inherited together. However, recombination events can occasionally separate them, proving that they are distinct loci. Sometimes they are separated in some generations, so then we can understand that yes, they are different loci. They are not in the same locus, they are different genes. Okay, so what is genetic linkage, what is recombination, I'll come to this in later classes, but for now you have to understand that pseudoalleles are the alleles of different genes. They are just functionally similar, okay, and they are very closely located to each other. I hope you understood. So it helped illustrate that the classical concept of gene might not always apply. So as functionally related genes can be located in close proximity and behave if there are allelic variants. So functionally related genes may sit close together and they behave like they are alleles. Okay, they behave like they are the allelic variants. So the summary: the lozenge locus in Drosophila is a classic example of pseudoallele, where a cluster of close related genes governs a related function, that's eye development, and mimics the inheritance patterns of alleles in a single gene. They mimic that they are a single genes. They are a variety of different alleles, but they are completely different genes. Okay, where a cluster of close related genes governs a related function, that's eye development, and mimics the inheritance patterns of a single gene. Likewise, a single gene's alleles all are inherited together. They mimic that situation, but they are closely related genes. There are many genes that govern the same function, that is eye development. I hope you understood what is pseudoallele. Pseudoalleles are nothing but the alleles of different genes. They just lie very close together, mimicking the allelic variants of a same gene. Okay, now we will study isoalleles. Okay, so isoalleles are alleles that are nearly identical in function and genetic sequence, but may show minor differences in their phenotypic expression. Isoalleles generally produce the same or very similar phenotypes, but small variations might exist in the intensity or expression levels. Okay, so what's the difference between isoallele and pseudoallele? In case of pseudoallele, they are of different genes, but in case of isoallele, these isoalleles are the variants of same gene. Okay, they are the variants of same gene. Here you can see the white gene in Drosophila that is responsible for producing pigments of the eye, resulting in normal eye color, normal red eye color of the fruit flies. But here you can see so many red eyes, from dark red to pink. So many type of eye color are there. So this is the variation of a single gene. Okay, the variation of a single gene can produce so many phenotypes. So many variation of the genes are known as isoalleles, and when there is a mutation in this gene, they produce white eye, that is known as white eye mutation in Drosophila. So this white eye gene is very much important. These are the variations of this white eye gene. This is the variation of the white eye gene. So isoalleles are the alleles of a similar gene, just a slight variation of the similar gene. Okay, a mutation in the white eye gene causes a complete loss of pigment, leading to the characteristic of white phenotype. We have witnessed that. So I hope you understood what are isoalleles. Isoalleles are similar genes. Isoalleles are similar genes, thus the similar genes are modified in some way and they are producing different phenotypes, but pseudoalleles are of different genes. Okay, so in this case, the white gene isoalleles could exist as slightly different versions of white eye mutation, where some flies might have very pale red to light pink eye color instead of a completely white eye. Okay, this is due to slight differences in the gene sequence or in the regulation of the white gene. That is the main case of the isoallele. These isoalleles do not result in completely normal red eyes but display phenotypic variation in the degree of pigment loss, possibly due to partial function or regulatory differences of the white gene. Actually, this white gene, that the gene responsible for production of the pigment. Okay, there are some variation of this pigment production in different, the genes changes their sequence to give rise this very type of phenotype. For example, I'm giving you a very lucid example: capital W producing totally red, capital W, small w producing pale red. Okay, there are lots of combinations: small w white. Okay, so like these, there can be, I just gave you two variation. There can be lots of variation in the same gene, so production of lots of types of alleles of the same gene. That is why there are very type of phenotypes in the eye. From red eye to pink eyes, there are lots of types of variation. So the same gene can have different types of alleles because of the difference in genetic sequence. So these are known as isoalleles. I hope you understand what are isoalleles. So I have given one very important thing, that the difference between isoallele and pseudoallele. I hope you may wonder what are then difference. So the first difference I already told you, that the isoalleles are from similar gene and pseudoalleles they are from different genes. So slight variation of the same gene of isoallele, but the pseudoalleles closely linked but they are distinct gene. In genetic basis, minor differences within the gene. There are isoalleles are minor differences in the gene, but they are separate gene behaving like alleles. In case of pseudoalleles, they are separate genes but they behave like alleles. Okay, phenotypic expression very similar or indistinguishable. Here you can see that the eye color of the Drosophila, there are lots of type of red variation, but very indistinguishable from each other. Okay, so that is the case of isoallele. But in case of pseudoallele, similar but can show a distinct trait. They function in a similar way but they can show distinct trait. Okay, mapping, isoalleles are of same locus because same gene, they will be from same locus only. In case of pseudoalleles, they are of different loci because they are different gene but they are closely linked. I have shown you the picture that the two alleles are closely linked but they are of different loci because they are of different genes. Example, white eye mutation isoallele I showed you, and in pseudoallele, lozenge locus pseudoalleles. And recombination not relevant because they are similar gene. Between similar gene, how can recombination happen? But pseudoallele, because they are different genes, the recombination is possible, but it's very rare as because they are located very close to each other. Okay, so isoalleles are variants of a single gene, which I told you that they are only the variants of a single gene, while pseudoalleles are different but closely linked genes that can mimic allele-like behavior. Pseudoalleles are different genes but they mimic like they are alleles. Okay, so isoalleles are the variants of a same gene and pseudoalleles are the different genes but they mimic allele-like behavior. Okay, I hope you understood what is isoallele and what is pseudoallele. You may wonder then, ma'am, what is the difference between multiple allele and pseudoallele? I will tell you that in case of multiple allele, for ABO blood type, there are three distinct alleles. There are no variation of the alleles, but in case of isoallele, for example, the gene beta actin. I'm just giving you a very arbitrary example, beta actin. The genes, there can be different types of allele, small a, capital A, small a, plus, small a, capital A plus. So there can be different variation of the allele in case of isoallele, but for multiple allele, there will be no variation of the allele. There are three distinct alleles present. In each individual, there will be two alleles, but in population, there can be three distinct alleles. But there will be no such combination of the alleles. Okay. But in case of isoallele, there can be so many variation of the allele. So many genotype can be represented by this type of allele. So many variation of the alleles are there. But in case of multiple allele, no variation, only three distinct type of allele present. When three distinct allele are present in this type, they can produce different blood type, that is AB, AA, B blood type, O blood type. Just very simple. But in case of isoallele, allelic variants are high. There can be so many variants of alleles that from red color to pink color we can get so many variation. Okay, so this is the difference between multiple allele and isoallele. Now coming to the last part, last variation, that is the lethal allele. It's very important. A lethal allele is a type of genetic mutation that leads to the death of an organism when present in specific genotype. It's very important. Lethal allele, when present in a specific genotype, can cause death. Okay, these alleles disrupt essential biological processes. By the name you can understand lethal allele. Lethal means death. Okay, they can affect many biological processes, making survival impossible under certain genetic conditions. Lethal alleles can be dominant or recessive and are often associated with developmental defects. They are often associated with developmental defects, metabolic failure, structural abnormalities, and they can be divided into two, that is dominant lethal and recessive lethal. Dominant lethal means in a single dose also it can die. Okay, you know from the dominant concept that one dose of dominant allele can cause the phenotype. So if two capital A present, they will die. Also if one dose is present, they will also die because they are lethal allele. Lethal means death. And in case of recessive allele, what you can see, in case of recessive allele, small l will die, because in order to become the recessiveness expressed, you need two dose of the recessiveness, small l, but if capital L small l, they will survive. Okay, only small l will die. So what is dominant lethal allele? These allele cause death even if only one copy present. I told you that capital L small l, only capital L is enough for death. That is in heterozygous individuals also. Dominant lethal alleles are rare because they tend to eliminate individuals before they can pass on the allele to offspring, because they themselves die. How can they pass their alleles to next generation? Okay, example is Huntington disease. It's very important. In humans, it is an example of dominant lethal allele. It's a neurodegenerative disorder, okay, that is caused by a dominant mutation, but the lethal effects typically manifest later in life after the individual has had the chance to reproduce, and at that time they notice that they had this lethal condition, okay, that is Huntington's disease. Now the recessive lethal allele. These alleles only cause death when an organism is homozygous for the lethal allele, that it inherits two copies of the allele, okay, one from each parent. I told you that small l will die, but in heterozygous individuals the lethal allele is masked, allowing the organism to survive. We know capital L small l will survive, but small l will die. An example is numerous, that in mice the yellow coat color allele, that is AY, is dominant for color, but it's recessive lethal, that mice that are homozygous for the yellow allele, that is AY die during early development, while heterozygous allele AYA survive and show the yellow coat color. Okay, so AY, this is dominant for color, but if this is present in double dose, it is a lethal combination. It is recessive lethal. Okay, I hope you understand. The thing is that if AYA present, the coat color will be yellow, but they will survive because this is dominant. Okay, this is dominant for color, but if they are present in double dose, they will be yellow and at the same time they will die. So this is dominant for color, but this is recessive lethal. When present in double dose, it is a lethal condition. Now another example of lethal allele, that is Manx cats. This Manx gene is associated with taillessness. It's a dominant lethal allele. Cats with one copy of the allele have a shortened or absent tail, but those who have two copies, homozygous, die before birth. Okay, here you can see that two Manx cat MK capital M, small m, capital M, small m, when they are crossed, you can see that capital M early death, capital M, small m max, capital M, small m max, small m non-max. So this capital M, this gene is a dominant gene, dominant lethal allele. It's a dominant lethal allele. When present in single copy, the cats have no tail, that is known as taillessness, and when present in double copy, that is capital M, then the cat die before their birth. Okay. And another example of lethal allele, that is sickle cell anemia. I have already given you the example in previous class, and that in humans the sickle cell allele can be lethal when present in homozygous state, and that the small a, that's a lethal condition. Okay. Capital A, small a, capital A, small a, they are heterozygous individuals. When they are crossed, one capital A, it is normal, one capital A, small a, they are carriers, and small a, it is sickle celled. Okay, these individuals have sickle cell anemia, and it's a lethal condition. But these heterozygous condition, they get an advantage from malaria, as malarial parasite do not affect the sickle celled red blood cell. Malarial parasite need the full healthy round red blood cell, as these heterozygous patients, they have some round red blood cell and some sickle celled. So the plasmodium do not tend to affect these individuals, and that is why these individuals get advantage from malaria. So they are not sickle celled, at the same time they are saved from malaria disease. So that is also known as heterozygous advantage. Okay, as because they are heterozygous, they are not sickle cell, they are the carrier of sickle cell anemia, because one small a is there only. If two small a is there, then they are the patient, there is one small a, so they do not have the sickle cell anemia, as well as they are protected from malaria. That is also known as heterozygous advantage. Okay, one last thing for lethal allele, also known as conditional lethal, that is that some individuals are environmentally lethal, that some alleles are conditionally lethal, that they are okay in some environmental condition, but upon change of the environmental condition, the situation becomes worse. For example, an organism might survive with a lethal allele under normal conditions, but die if exposed to a certain temperature, chemical or other environmental factors. This may also happen. Okay, so in summary, a lethal allele is a genetic variant that causes death when present in a particular genotype. So the genotype is very important for recessiveness. Presence of two recessive allele important for the death. For dominantness, only one dose is also enough for the individual to die. So a particular genotype is also important. A lethal allele is a genetic variant that causes death when it is present in a particular genotype. These alleles can be recessive or dominant and often disrupt vital processes. That is why they are really lethal. Why they can be lethal? They are lethal because they are affecting the vital human processes, the metabolic processes, the vital processes that are regulating our body. Okay, so they are highly impacting our lives. That is why they are known as lethal alleles. I hope you understood, students, what is pseudoallele, what is isoallele, and what are lethal alleles. If you have any queries or any questions, you can comment below, and if you like my videos, you can subscribe my channel and press the bell icon for the update. Next class I'll come up with more genetics lessons. I wish you happy learning, students. Thank you so much. All the best to all of you.